and hope you had a very, very beautiful week. Today, we are going to continue our lectures on serologic techniques or serological tests used in the diagnosis of infectious organisms. And last class, the main test we saw was the rapid diagnostic test, which have rapidly gained their way into most laboratories today. Of course, we highlighted some of the advantages of those tests being used as point of care, the no need for use of expertise technicians and so on. Today we're getting to more complicated procedures, and these ones will always need a trained technician to run. Of course, there are still some simple procedures, but I'll always indicate when it is possible. Remember, last class I mentioned that serological tests are classified as primary, secondary, tertiary, depending on the concept. A primary test, as I indicated last class, measures the interaction between measures the interaction between an antigen and an antibody in vitro, that is, inside a test tube. This could be targeting a virus, a bacteria, a fungi, or a parasite. Secondary test, on the other hand, measures the implication of this binding between antigen and antibodies. And this could be observed through what? Agglutination, precipitation, or complement fixation. And tertiary, as I indicated, it is in vitro. That's the only in vitro test, which measures a consequence of the interaction between antibody and antigen, which could now result in neutralization of the antigen in the system, and you find the person recovering. We'll be talking mainly on primary and secondary tests. We are not going to look at a tertiary test in this course. But today, we are going to look at primary tests. We are going to exhaust all the primary tests. So our advisor, you get a good seat. And if your data is running out, try to update. Talking of primary tests, these are the different primary tests that we are going to look at today. Enzyme-linked immunosorbent assay, ELISA. We look at radioimmunoassay, Western blotting, Southern blotting, Northern blotting, and fluorescent test. The list may look too long, but I'll focus so much on ELISA and fluorescent test. The other ones, I may have just a slide or two, because in reality, radioimmunoassay is really limited in application because of radioactivity, as you already know. Western blotting, Southern blotting, and Northern blotting are restricted to sophisticated laboratories. So most likely you'll be working with ELISA or fluorescent test. That's why I'll focus a little bit more on these two. Okay, I think with that in mind, we can kickstart the show. So we are going to begin with immunofluorescent test. And there are two types of immunofluorescent test. We have the direct and the indirect. The word direct and indirect depends on what is being labeled. Okay, if we are labeling directly the antibody that is reacting with our antigen, we call it a direct test. If you are labeling another antibody that will now bind to our antibody antigen complex, we now call it an indirect test. So that's where the word direct and indirect stems from. So we'll begin with direct fluorescent antibody test. And I want you to pay attention in this because this is almost a similar technique that we'll be using in ELISA. So it is used to detect and identify unknown antigen in specimen. So saying that you have been infected with an organism, it could be a bacteria, a parasite, a virus, or even a fungi. as we indicated in the first lecture on this section, when you are infected, the parasite wholly provokes your system, and your system secretes antibodies which are specific to the parasite, mostly in the case of viral and some bacterial infections. Or on the other hand, they release some proteins. They release some components of their system, usually surface proteins in the case of viruses, or surface components in the case of bacteria, or polysaccharides, lipopolysaccharides components. And these components provoke an immune reaction, and the immune system then mounts a response. When this response is mounted, when this response, a reaction between the antibody that has been system and result in the neutralization of antigen. Results in the neutralization of the antigen. And the infection is subsided, or you can be, the infection is controlled. So in the indirect test, as I indicated earlier, the fluorescent dye is attached or labeled directly to the antibody that is specific to the antigen we want to test. And we can now observe this binding under a fluorescence microscope. I'm sure with Dr. Moka and Dr. Lem, you have been drilled on the different types of microscopes. So you understand the principle of fluorescent microscopy very well. So this is the procedure. A tissue smell or a tissue sample of blood or serum is fixed to a microscope, a slide. I will soon start taking notes of students who make noise in my class. And be careful with what I will do with you. Battery Renee. So, as I was trying to indicate the procedure, the antigen could be in your tissues, could be in your blood, or anywhere in your system. We obtain that sample that we think contains your antigen. We fix it to a slide, and then we label an antibody with a fluorochrome, any fluorescence component. This labeled antibody is now allowed to react with our antigen. So we pour our labeled antibody onto our slide that was fixed with our antigen. Antibody-antigen reaction, as I already stated, will occur, and we will wash the system to remove any unbound antibodies. Remember, these antibodies are labeled. So if they remain in the system, we are going to observe their fluorochrome, and which will not mean that there is a positive reaction. So when we wash, we'll remove all the unbound antibodies. And when these unbound are removed, we'll intimate that any labeled antibody found in our system is an antibody that bound to an antigen. It's an antibody that was bound to an antigen. Therefore, when we observe under the fluorescent microscope, any fluorescence that we observe is a positive test. If there was no antigen on our sample, if we did not fix any antigen in our sample, the antibody that is labeled will have no binding site on our slide. And therefore, during the washing stage, every labeled antibody will be washed, and when we go under the fluorescent microscope, we are not going to see any. We are not going to observe any fluorescence. There are so many different uses of this fluorescence antibody tests for bacteria very low concentrations. It may also be used to dictate viruses growing in tissues and in cultures, and especially the HIV antigens. So it's really used in dictating HIV antigens. Indirect antibody test is the next one, as I already indicated. Again, the procedure is the same. It begins the same. We fixed an antigen to a slide. We allow this antigen to combine with an unlabeled antibody. So we incubate our antigen with an unlabeled antibody. We wash the slides. Remember, washing again is to remove any unbound antibodies. Then next, we add a labeled antibody. So we are adding an antibody that will bind to an antibody to the system. The first antibody bound to our antigen, that was a specific antibody to the antigen we wanted to test. Now, the labeled antibody is going to bound to our bound antibody. So if the antibody did not bind to our antigen, when we add the labeled antibody, there will be no binding. And in this second washing, we are going to wash all labeled antibodies that are unbound. And at the end, we'll go and observe under the fluorescence microscope for fluorescence. So if we see any fluorescence, what would that mean? It will mean that our unlabeled antibody bound to the antigen, and the labeled antibody bound to the unlabeled antibody, and finally we're able to see that fluorescence. If the antigen is not present, the specific antigen is not present, the unlabeled antibody will be washed, since it will not bind to anything, will be washed in the first step. And when we add the labeled antibody, the labeled antibody will also be washed in the second step, the second washing, since they're still not binding, and therefore we will not see any fluorescence under the microscope. As usual, you can interrupt me with a question or interrupt me to retake it over if it is not clear. So it can also be used to dictate antibodies. Remember, so far I've been explaining about using it to dictate antigens, but we can also use it to dictate antibodies. In this case, we just reverse the what is bound to the slide. So we'll bind first, but the antibodies to the slide, and then now we apply what? The unlabeled antigen. And secondly, we now add the labeled antibody. So the antigens will now have two binding sites to antibodies. The first will be the adsorbed antibody, and the second will be to the labeled antibody. So at the end, we are going to see fluorescence if there is any binding. And if there's no binding, as I explained earlier, there won't be any fluorescence. So this is a procedure. You can take time and read it. And the uses, of course, almost the same. It's just the principle, what is being bound at the first step that will change. That is fluorescence antibody test. We have looked at the direct and the indirect. Now we go to the second method. This method is more popular because it is commonly used. Enzyme-linked immunosorbent assay, ELISA. You always say the word ELISA, not enzyme-linked immunosorbent assay. But as a scientist, you should understand that that is what it means, and it is used in the diagnosis of microbial infections by detecting the presence of antibody or antigen, just like in the indirect fluorescence antibody test, where either detecting an antigen or an antibody. It is the same thing with ELISA. So I'll always indicate when you can use it for antibodies, when you can use it for antigen. So it is all about the steps. What do you bind in the beginning, just as before? ELISAs are very specific and very sensitive and are required only in very small, so can dictate very small amounts of antigens or antibody in specimen. So immediately we start producing antibodies, ELISA is able to pick it even within the first few hours or first few days. The reagents used in ELISA are also very stable with a long shelf life, meaning that we can send them to even district laboratories and they will be able to stand the test of instability in terms of climate and even without electricity. So that is a plus for it. And then finally, it has been used either in diagnosis or in industries. So we can either use ELISA to diagnose infections or we can use ELISA for quality control in industries. Again, it has a very high sensitivity and high specificity and can be used in high throughput. So we can measure so many samples almost at a go. So, and a typical ELISA plate is a 96 well. This is a 96 well plate. So each well will be a sample on its own, meaning that at a point we can measure, we can test 96 different patients at a time, but it will not be 96 because we always have what we call positive and negative controls. So, or we can, let me say we can test up to 90 samples at a time. Meanwhile, in other tests, immunofluorescence is one sample at a time, mostly one sample at a time. So ELISA has the high throughput capacity. In its typical principle, an unknown amount of antigen, of course, it could be an antibody, is fixed to a surface. And then a specific antibody is washed over the surface so that it can bind to the antigen. That is when we want to test for what? Antibodies. On the other hand, we can use an unknown. So let me take the first case. An unknown amount of antigen is fixed to a surface, and then a specific antibody is washed over the surface so that it can bind to the antigen. In the other case, an unknown amount of antibody is affixed to the surface, and then a specific antigen is washed over the surface so that it can bind the antibody. So as I said, it all depends whether you want to test for antibody or test for antigen. What do I mean here? You collect specimen from a patient. Do you want to test for antibody in that specimen or you want to test for antigens in that specimen? That is the whole story I'm trying to tell here. So in the specific case of antigen, the antibody is... So let me go through with the diagram. This is an antigen. This antigen is fixed to a microtetra plate. A microtetra plate. This antigen is fixed there. A fixed amount of antibodies is added. These antibodies are added. It is allowed to react. It is allowed to react. Then a labeled antibody and this antibody binds to the primary antibody. It's a primary antibody. And then we observed yes. Okay, Chess Desmond from one. Okay, sorry for the mix-up. I'll take it all over. I'll take the principle of ELISA all over because it's very important, as we are going to have 3 different types of ELISA to look at: indirect, sandwich, and competitive. And the basic principle must be well understood. So in the typical principle of ELISA, an antigen is fixed to a microtiter plate. This antigen concentration is unknown. Next, a primary antibody of known concentration is allowed to react with this antigen. A primary antibody of known concentration is allowed to react with this antigen. Next, a secondary antibody is conjugated to an enzyme. A secondary antibody is linked to an enzyme. The word enzyme linked aminosorbent assay. A secondary antibody is linked to an enzyme, and this secondary antibody is allowed to bind to the primary antibody. Next, a substrate to this enzyme is added in the mixture. A substrate to this enzyme is added in the mixture. And what happens last? If there was a reaction, this enzyme will break down the substrate and will be observed by a fluorescence, a color change. So the substrate is usually colored so that when the enzymes breaks down the substrate, the enzyme substrate reaction releases a color change which can be detected. The enzyme substrate reaction results in a color change, and the dictation of this color change is a positive test. If this antigen is not specific to this antibody, if this antigen is not specific to this antibody will not bind to the antigen. So the primary antibody will not bind to the antigen. And when we add the conjugated antibody, there will be no antibody, there will not be no primary antibody for the conjugated antibody to bind, and therefore this man will not bind. And when we add a substrate, the enzyme is not present, and there will be no color change. Ni Priska. If I take that, name is -3 marks. Okay, so I've gone through it, but this is just for reading. That in this technique, non-antigen is attached to the inside surface of the well, and patient serum is added. After incubation and washing, enzyme-labeled anti-human globulin is reacted with the antibody that has attached to the antigen. The presence and concentration of antibody that has reacted with the antigen is shown by a change in color. When the substrate is added, the intensity of the color is directly proportional to the concentration of the serum. So these are the steps. You take your time and you read the steps, but I'm going to explain again using the typical diagrams for indirect ELISA. Remember, I said there are 3 different types of ELISA. We have indirect ELISA, you have competitive ELISA, and you have sandwich ELISA. So the indirect ELISA is the ELISA I just explained, but let me go through the procedure steps, which I just skipped the text because I think it's more better to observe with the image. So this is our container. Okay, this is our microtiter plate. This plate here. Always have this in mind when I talk of a microtiter plate. This is what I'm referring to. That's why I brought this image here so that I have an idea. It's usually 96 wells. So we could be using just one well, or we're using all the 96 wells. It all depends on whether we have how many samples to test. In reality, most ELISA plates come with an antigen or an antibody already absorbed to it. In reality, most ELISA plates come with an antigen or an antibody already absorbed to it. So let's continue to understand that we want to test for antibody in the patient's sample. Want to test for antibody in the patient's sample. So we absorbed a known constitution of antigens. We absorb antigens, a known antigen, onto a microtetra plate. So in practical sense, we want to test for HIV antigens. So the HIV antibodies in patient serum. We want to find out if this patient has HIV antibody that is seropositive for HIV. So we are going to absorb HIV antigens to this microtetra plate. When we absorb the HIV antigen, we incubate. Incubate is allowing time for it to stick, to fix to the walls of the microtetra plate. So when it becomes fixed, you'll find it like this. It will be all attached. So we can wash the mixture to make sure that any unattached substrate is removed. Any unattached antigen is removed. So this is what we are going to get after washing. Now, as I said that in real life function of ELISA, these plates already come at this stage. Okay, ELISA plates already be at this stage. We have antigens already adsorbed to the walls of the plate. So after adsorption, we block with a bovine serum albumin. This is a non-specific protein to ensure that there's no space left in between the attachment so that our antibodies does not unspecifically bind to the surfaces of the walls of the plate. So that's what we block. After blocking, we introduce our specimen. It could be blood, it could be plasma, it could be serum. Any of these three will do the job. But generally, we encourage that you use serum. So you include you include the serum of the patient you want to test for HIV. If HIV antibodies are present in the patient's serum, meaning that the patient is seropositive for HIV, those antibodies will bind to the antigens, to the HIV antigens that are adsorbed to the walls. Those antibodies are going to bind to the HIV antigens that are adsorbed to the walls. After this binding, we incubate. After incubation, we wash again. That washing is to remove any unbound antibodies so that we don't have antibodies floating inside our mixture which are not bound. Next, an enzyme conjugated antibody, an enzyme labeled antibody is introduced. This antibody is expected to bind but to the bound antibody or to the antigen antibody complex. So after this, we wash again. This washing is to remove any of these antibodies that are not bound to our complex. Finally, we add a substrate of this enzyme. The substrate of this enzyme will bind to this enzyme, and the enzyme will break down the substrate, and it will result in a color change. The enzyme will break down this substrate and result in the color change. If we want to just know whether the antibodies are present or not, we are going to simply observe color change, which is an indication of antigen-antibody reaction. But if you want to know how much of the antigens, sorry, how much of the antibodies are present in your system, we'll have to go to what we call quantitative reading using an ELISA reader. So we'll go to a spectrophotometer to quantify the intensity of the color. The more, if we had all these antigens occupied with antibodies, it will mean that we'll have more of these substrates available for the enzyme. So the more of these enzymes available, and therefore more substrate will be broken down, and the intensity of the color will be very high. If it was less, as we're seeing now, we have just two antibody binding, we have very less intensity of the color. In some cases, we can dilute. As we move from one dilution factor to the other, we observe the color intensity. That will also be quantitative ELISA. I go now to sandwich ELISA. Remember I said you can always stop me if a step is not clear. I go now to sandwich ELISA. The principle of sandwich ELISA is almost the same as the principle of indirect ELISA, but it specifically measures antigens. It is used to dictate antigens in sample. So in this case, the plates are coated with what? Captured antibodies. The plates are coated with captured antibodies. The sample is then added. The sample is now added to the captured antibodies. Of course, I will not be repeating that wash, because you already know that after every step, you have to wash to remove the unbound. After every step, you have to wash to remove the unbound. So when you add the sample containing the antigen, the antibody is going to capture the antigen. So I'm going to evict Ni Priska from the class. I don't know why for the second time she's unmuting herself when she's not even asking a question. So I'm going to remove her from the class. Meanwhile, she will be penalized as already indicated. Let me go over again for the sake of the distraction. Sandwich ELISA is having almost the same principle like indirect ELISA, but the first major difference is that we want to measure antigens and not antibodies, as we saw in indirect ELISA. Here, we are going to absorb antibodies onto the slide. As I indicated, in typical cases, those slides already come with those specific antibodies adsorbed to them. But let's assume that we don't have those slides and want to prepare the slides ourselves. Sorry, the Zoom people, I will have to restart Zoom because that's how it works. Okay, I think it's good to pick up again. We're on sandwich ELISA. Please, Skype, can somebody unmute and confirm that you see my screen?
Yes, sir, we can.
No, sir, I'm not
can
seeing
see your screen.
Okay, good.
I'm not
Those
seeing
are
your
not
screen,
seeing.
sir.
If you are not saying sign out of the call and rejoin the call again, you have network issues. That's why you cannot say sign off of the call and rejoin the call again. So we're on sandwich ELISA, and we already passed the stage where you have captured antibodies onto the microtiter plates, you are washed, and then you block, and then the sample, the serum from the patient, is added onto the captured antibody. If there is a specific HIV antigen in this patient's serum, that antigen is going to bind. That antigen is going to bind to the antibody. If the antigen is present, the antigen will bind to the antibody. Now, an enzyme-linked antibody, a conjugated antibody, is introduced. That conjugated antibody binds again to the same antigen. That conjugated antibody binds again to the same antigen. So you have the antigen sandwiched between two antibodies. That is why it is called sandwich ELISA. You have the antigen sandwiched between two antibodies. And then finally, the substrate of this enzyme is then added. The substrate of this enzyme is then added. And then just as in the direct ELISA, the enzyme will break the substrate and will observe for color change. In some cases, this secondary antibody is not conjugated to an enzyme, meaning that we will now add again another antibody, which is an anti-antibody to this antibody, which is now conjugated to the enzyme. So, but don't get mixed up. A third antibody may be used, a third antibody might not be necessary. It all depends whether the secondary antibody is conjugated or not. But in the typical explanation of the procedure, you could eliminate that third antibody and end at the fact that an enzyme conjugated antibody is added, a second antibody is added, which now captures the antigen at another epitope, and the antigen is therefore sandwiched between the two antibodies. And you can now add the substrate and observe for color change. The next is competitive ELISA. Moise fidelity. Okay, competitive ELISA. This is the third type of ELISA. And as the word implies, there is competition. There is competition. So by default, this ELISA is a quantitative ELISA. Competitive ELISA by default is a quantitative ELISA. So the first thing we do is that an antigen is coded to a well. Forget about the order of these steps. An antigen is coded onto a well. A known concentration of antigens is coded onto a well. The patient's sample is allowed to react with another antigen. It could be the same antigen. I think typically it is the same antigen. So we take a patient's sample, blood, serum, we allow to incubate, so we mix it with a particular concentration of antigens. We know the concentration of antigen, but we don't know the concentration of the antibodies in the patient sample. So we allow the patient sample to react with this antigen. After this reaction, we now take this mixture and introduce into the coated plates.
The stone heart that will be having now.
The antigen antibody mixture that was added, free antibodies, that means we bind to the antigens in the coated plates. Yeah, lucky this time that I could not identify exactly who was causing the noise. So what I'm saying is that we have coded a particular antigen into a plate in non-concentration. We have used that same antigen to react with the patient's serum. So the first step is, after coding, we allow outside the microtetra plate for incubation between the antigen and the antibody. When we now pour this interaction into the coated plates, we expect that only free antibodies will be available to bind to antigens in the coated plate. Only free antibodies will be available to bind to antigens in the coated plate. After a second incubation, we wash. After washing, we now bring a secondary antibody, which is specific to the primary antibody. This secondary antibody, of course, is conjugated to an enzyme. And then the substrate is added for a reaction to be observed for a color change. Pay attention. Let me explain what we're trying to do here. This is the same antigens. You see the color is the same. These antigens have been coded here. These same antigens have been allowed to react with these antibodies. Now this mixture, these antibodies are coming from the patient's system sample, of course. This mixture, all of this is now poured into this. What does that mean? That only free antibodies will be able to bind to coated antigens. Only antibodies that are not bound to the antigens outside the coated plate will be able to bind to antigens in the coated plate. And then when we wash, mean that we're going to wash this off. We're going to want the antibodies, antigen complex that reacted outside of the coated well off, and only the antigens, antibody complex that are coated onto the well will be left. And when we add the substrate, we'll observe for a signal. The higher the antibodies in the patient's system, the higher the antibodies in the patient's system, the higher free antibodies will be available after this primary incubation. Meaning that the more antibodies will be available to bind to coated antigens, meaning that the higher the intensity of the color. The higher the intensity of the color. If we had only few antibodies present in the patient sample, all those antibodies, or just one or two, will be left after this primary reaction, meaning that we'll have just few antibodies reacting with the coated antigens, and therefore a lesser color intensity. If we have a very, very few number of antibodies, it means that we'll not even have excess of these antibodies to be available to react here. And therefore, we might not even have a color intensity. So competitive ELISA is a quantitative procedure to dictate the concentration of antibody by competitive binding. Again, you must be able to link to me how the color intensity is related to the concentration of antibodies in the patient's sample. You must be able to link that. Let me take it all over. If we have more antibodies in the patient sample, it means that after this competitive binding, we still have enough antibodies to bind to coated antigens in the well, meaning that more antibodies will be available to bind a conjugated antibody, and more conjugated antibody will be available to break down the substrate, and the higher the signal. The reverse, of course, is true. Radioimmunoassay. I don't pay so much attention to this as I indicated earlier because of the limited applicability, the limited use in diagnostic procedures. Of course, it has a wider applicability in other settings, but in diagnostic procedures, it has a very limited applicability. It is a competitive process, and it measures radioactivity. Unlike the other ELISAs and flow, you cannot afford to be texting me. I've said you can unmute yourself and ask a question. A student is asking, does it mean the color intensity depends on the concentration of antibodies? Yes. The higher the antibodies available, the higher the bound antibodies, the higher the conjugated antibody, and the higher the color intensity. We have very few antibodies available. It means just few will bind here, and so very few will be able to bind. Since we wash this complex off, wash these ones off, so only this one will be able to bind. And if there are few of them, it means the conjugation will be less, and the substrate binding intensity, substrate breakdown intensity will be less. So avoid texting if you want an immediate response to the answer. So different radioactive isotopes have been used. We have tritium, carbon-14, sulfur-35, phosphorus-30, and iodine-125. These are all different isotopes. So these isotopes are used to label antigens or antibodies, depending on what we want to test. These isotopes are used to label these antigen or antibodies. And after a reaction, just as we've described, we now test for radioactivity. After a reaction, we test for radioactivity. The presence of radioactivity is an indication that the labeled isotope was present. The absence of radioactivity is an indication that the labeled isotope is not present. So as indicated, it's very sensitive, very, very sensitive. So common applications in dictation of hormones, drugs, testing, of course, enzymes and even microbial antigens, but as I said, it is very limited in application of diagnosis. So the next procedure is Western blotting. Western blotting. Western blotting is used to test the specific binding of protons. Western blotting is used to test the specific binding of protons and is more commonly applied in HIV diagnosis to test for the presence of P24 or P55 protons. So when we dictate the presence of these protons in samples, the technique is called Western blotting. The details of the techniques are not important for this lecture. A more widely applicable technique is what we call Southern blotting. And Southern blotting is named after the scientist E.M. Souding, who described the technique for the first time in 1975. Here he used specific DNA sequences to dictate mutation. So I'll talk a little bit about the principle of Southern blotting, because it is from this principle that we are able to explain Western and Northern blotting. So this is a DNA sequence. This is a DNA sequence. A double-stranded molecule. when we have this when we separate this DNA sequence, of course, you can use heat, you can use helicase. We have one sequence here and one sequence here. So if I use the same DNA molecules in two different tubes, I separate them in two different tubes. So tube A has this strand. So let me take it all over. This is my original DNA molecule. I separate. This is my original DNA molecule. I separate it into two. When I separate this DNA molecule into two, I put this separated as I make sure that the separate molecules are actually well apart. So I bring tube A. This is tube A. This is tube B. So tube B has all of this, and tube A has all of this. If after some time I allow this to react again under permissive conditions, appropriate temperatures, in particular pH and the rest, I'll expect that tube A will bind back with tube B. I'll expect that tube A will bind back with tube B. Now, let's talk southern blotting now. I want to find out if you have the DNA of Salmonella in you, for instance. I want to find out if you have the DNA of Salmonella in you. So I have my Salmonella DNA that I have separated. So this is my Salmonella DNA. In my tube, and then this is Salmonella DNA in sample. If after mixing these two, there is hybridization, I can confirm that this one and this one were related. If there's hybridization after mixing these two, I can confirm that this and this were related. But if there's no hybridization, I'll say that what? This man and this man are not related. If there's no hybridization, I'll say they are not related. Sounding, in particular, wanted to dictate mutations in the DNA molecule. So I could take the same Salmonella DNA molecules from two different strains of organisms, or from two different samples, but I know that both of them are tested for Salmonella, are allowed for them to bind. If there's sufficient mutation in this one, there will be no binding. So binding is implying there is relationship. Lack of binding could mean that one, there's no relationship, or two, there's sufficient mutation to prevent binding. So if we are doing this in protons, it is called Western blotting. If we are testing this to ascertain the presence of protons, Western blotting. And if we are testing this in RNA, it is called Northern blotting. So with that, next class we'll look at secondary binding and explore different techniques that employ secondary binding. I end the lecture